Bismillahirrahmanirrahim, and welcome back to this next video. And this is the third video in the series of videos on the insulin signaling. In the first video, we talk about the insulin receptor in detail, about its important subunits, that was the alpha and the beta subunit. And in my last video, we talked about the mature insulin, how the insulin, that is actually the signal that is produced, and the mature insulin that contain the A chain and the B chain, and they are connected to each other by the disulfide bonds at different positions of the cysteines. They contain the intra-chain bridges. Then you have got the interchain bridges between the A and the B chain to give you the mature insulin, which actually is responsible for binding to the ligand. Now, in this particular video, I want to start on the pathway, how the pathways work. Now, you have understand the receptor, its structure. You have understand about the signal, which is the insulin. So, how the insulin, which is the signal, that interacts with the receptor to give you the ultimate result of the glucose uptake, for example, by a cell. So, let us start about the insulin signaling pathway. Now, when the insulin that is produced, that actually travels through the blood, so it is using blood as a media, and when it travels through the blood, it is going to bind to the insulin receptor present on the target cell. Now, the insulin receptor, if you remember, I've told you that is a tyrosine kinase receptor. What I mean by the tyrosine kinase receptor is that this is a receptor and it have got an intrinsic kinase activity, and in this particular case, the kinase activity, that is a tyrosine kinase. What I mean by that is that this kinase, when it is going to act on its substrate, it will add a phosphate group on the tyrosine amino acids in its substrate protein. So as this is part of the receptor and this is a tyrosine kinase, therefore we call the insulin receptor as a tyrosine kinase receptor. So the insulin that is going to bind to the insulin receptor. Now, where does it bind? If you remember the last video, I've told you there are beta subunits and there are alpha subunits in the insulin receptor. So, the insulin binding site that is actually present in the alpha subunit. So, these are the alpha subunit, and the insulin molecule that is going to bind to both of them, to both of the alpha subunits. Now, what are the target cells? What I mean by that is, where are these insulin receptors present? You are going to see these insulin receptors on the muscle cells, on the adipose cells, and you're going to see them on the liver cells. So you're actually talking about the liver, the muscles, or the adipose cells where these receptors are present, and the insulin molecule, they are going to bind to them. So when the insulin molecules, they bind to the receptor, what happens? There are conformational changes in the insulin receptor, and these conformational changes are actually the signal that the insulin has bind to the receptor. We need to start the pathway. Now, when there are conformational changes, what these conformational changes do? These conformational changes, they are actually going to activate the intrinsic kinase activity of the beta subunit. If you remember in my last video, I've told you that these beta subunit, they actually have the tyrosine kinase domain, and they have the tyrosine kinase activity. So it is this beta subunit which is going to bind the phosphate group to the tyrosine. So the insulin binds, the conformational changes are taking place, and those particular conformational changes are responsible for the activation of the tyrosine kinase activity of these beta subunits. Now, what the beta subunit do is that when, because of the conformational changes, the tyrosine kinase domain of the beta subunit that gets activated, it is going to go for the autophosphorylation. What I mean by autophosphorylation is that this beta subunit is going to utilize its tyrosine kinase domain. It is going to add phosphate group on the tyrosine amino acid present in this beta subunit, as well as the tyrosine kinase, the tyrosine amino acid present in this particular beta subunit. So they are going for the autophosphorylation. They are not phosphorylating any other substrate at this particular time. They are phosphorylating itself on the tyrosine amino acid. Therefore, we call this is the autophosphorylation. Now, this autophosphorylation, what it do is that when it acts on the tyrosine, that would mean that you are going for the phosphorylation, you are adding the phosphate group to the tyrosine amino acid, you are creating the phosphotyrosines. Now, this phosphotyrosine or this autophosphorylation, what it do is that it activates the receptors and it creates the docking site for the intracellular signaling molecules that we are going to see in the next slides. By the way, who produces the insulin? It is the great pancreas, the beta cells or the islet of the Langerhans of the pancreas. They are actually responsible for the production of the insulin. So they produce the insulin, they move that into the blood. They move the insulin moves through the blood, bind to the insulin receptor on the liver fat or the muscle cells, and the story continues. Now, what I mean by phosphotyrosine. Now, this is the structure of a tyrosine. You may have seen the structure of the tyrosine. So you have got a hydroxyl group over here. Now, when it goes through the phosphorylation and you get the phosphotyrosine, as you can see over here, you are getting a phosphate group over here. And the addition of this phosphate group converts the tyrosine into the phosphotyrosines. Now, we have got the phosphotyrosines on the beta subunit of the insulin receptor. What happens next? Now, these phosphotyrosines on the beta subunit, they are actually providing a binding site for a protein which is known as the insulin receptor substrate protein. So when you talk about the insulin receptor substrate proteins, you're actually talking about a family of the signaling proteins. And in this particular family, there are 5 members in the IRS1, 2, 3, 4, and 5. And whenever you talk about the insulin signaling pathway in the context of the glucose metabolism, so the main players, they are the IRS1 and the IRS2. So these IRS1 and 2, they are going to bind to the excuse me, they are going to bind to the phosphotyrosines of the beta subunit of the insulin receptor. Now, what these IRS protein do is they play a central role in the intracellular signaling cascade initiated by the insulin receptor. So we have activated the insulin. Now that is going to activate the IRS, and these IRS, they are very important in the intracellular signaling cascade. Now, these insulin receptor, they act as adapter protein. What I mean by that is that they usually do not have any enzymatic activity. They are working as mediators. We call them as the adapter proteins. And what they do is that they are responsible for transmitting the signal from the insulin receptor to the downstream signaling pathways such as the PI3K or the AKT pathway. That is the focus of this particular series, how the glucose that is uptaken by the muscles, the fat, or the liver cells. But the insulin, they are also very important in the growth and differentiation of the cells, and that it do through the MAPK pathway that we will discuss in the videos to come after we complete the PI3K and the AKT pathway. So the IRS proteins, they are the adapter one, and they are actually responsible to mediate the communication between the activated receptor and the other proteins in the signaling pathway. So, what are the important domains that you see in the IRS protein? One of the important domains that you see in the IRS protein, that is known as the phosphotyrosine binding domain. Now, the name indicate this is a binding domain. Where does it bind? It binds on the phosphotyrosine. Now, this PTB domain is found at the N terminus, and what it do is that it is responsible for interacting with the insulin receptor after it phosphorylates on the tyrosine residues. You can simply say you have got the phosphotyrosines on the beta subunit of the insulin receptor. The IRS is going to utilize its PDB domain to bind to it. For example, if this is the beta subunit of the receptor, you have got the phosphotyrosines over here. The IRS is going to utilize its PDB domain to bind to the receptor, to the activated receptor. Now, the IRS, it has bind to the activated receptor. Another important domain that you see in the IRS, that is known as the central region. And the central region is actually providing a docking site for the SH2 domain containing signaling proteins like the PI3K that we are going to discuss in this particular pathway, and the GRB2, which is very important in the activation of the MAPK signaling pathway. So the central region is actually providing the docking site for those particular proteins which contain the SH2 domain, and the PI3K in the GRB2, they do contain the SH2 domain, so they can interact with the IRS protein utilizing their SH2 domain. Now, what the beta subunit do is, this is the beta subunit, they've got phosphotyrosines. The IRS has bind through the PTB domain. So when the IRS protein binds over there, what the beta subunit do is, it is going to phosphorylate the tyrosine residues in the IRS protein now. So the IRS that has bind to the receptor has the tyrosine kinase activity. Now this receptor is going to add phosphate group on the tyrosine amino acid of the IRS protein, and you are going to see the phosphotyrosines in the IRS protein now. Now, this phosphorylation of the tyrosine amino acid, or the creation of this phosphotyrosine, that is actually recruiting the IRS to the plasma membrane, and when that is recruited to the plasma membrane, it is going to enhance its interaction with the signaling molecules involved in the cell survival, growth, and metabolism. And when we talk about the glucose uptake, the recruitment of the IRS to the plasma membrane is responsible for the activation of another protein which is known as the PI3K, or which is known as the phosphoinositide 3 kinase. So you have activated the receptor, you have made phosphotyrosine in the IRS. The IRS is recruited towards the plasma membrane, and in the plasma membrane, it is responsible for the activation of the phosphoinositide 3 kinase, which for short is known as the PI3 kinase. Now, the phosphotyrosines on the IRS, that actually serves as the docking site for this PI3K. By the way, this PI3K is actually a lipid kinase, and you will see in a while why we call this is a lipid kinase. Now, this PI3 kinase, it actually have two important subunits. One is the regulatory subunit, which is known as the P85, and this P85 is going to bind to the phosphotyrosines present on the IRS protein. Then you have got the P110, which is actually the catalytic subunit, and this catalytic subunit is responsible for the kinase activity. So the IRS, utilizing its phosphotyrosines, interact with the regulatory subunit, that is the P85 of the PI3K, and that particular interaction is going to lead to the activation of the catalytic subunit, that is P110, that is responsible for the kinase activity of the PI3K. Now, what is the substrate of the PI3K? We have activated the PI3K. We have activated it for a purpose. We have activated it for the phosphorylation of something. And when you talk about the substrate of the PI3K, that is a phospholipid in the cell membrane, which is known as the phosphatidyl inositol 4,5 bisphosphate. For short, you can call it as the PIP2. It is sometimes also written as the PI4,5 in the braces, and then you have got the P2. So this is going to be the substrate for the PI3K. Now, as I've told you, the PI3K is a lipid kinase. It is going to act on this particular lipid to make to add a phosphate group over here. Now, this is how the PIP2 looks like. When you look at the structure of the PIP2, it looks like this. You have got two phosphate groups. That will be very much clear in the next slide. If you look at this one, we're talking about the cell membrane. We are talking about the PIP2. So if you look at the PIP2, it have got two phosphate groups at position number 4 and at position number 5. And therefore, we call this is the bisphosphate. So this is the phosphatidyl inositol 4,5. These are the position of the phosphate group. Bisphosphate, that means you're talking about two phosphate groups over here. When the PI3K that gets activated by the IRS, that is going to utilize ATP as a source of the phosphate group, that is going to convert that into the ADP, and it is going to take that particular one phosphate group and add that to the third position of the PIP2. What that means is that now you have the phosphatidyl inositol. Initially you have 4,5 bisphosphate. Now you have got 3,4,5 trisphosphate. Now you're talking about three phosphate groups over here. Now, when the PIP3 that has been made, what it do next is that will be the discussion of our next video. So we will continue the story in the next video.